So today we're actually going to be talking about the structure of the cell membrane. We've learned about the cell membrane last unit, so part of this should be review. So we're going to start with just the basic function, and that is it determines what enters and exits the cell. Okay, that's the first function. The second function, it really has to just do with animal cells, and it basically provides structure and support for animal cells. Remember, plant cells have a cell wall, and to be honest, the cell membrane doesn't do a really good job of providing structure and support for an animal cell, but it does provide some structure and support. It also obviously determines the shape of the cell because of that. The last function really kind of goes back to Unit 1. It's a word we talked about in Unit 1, and that is it maintains homeostasis. Okay, so what is homeostasis? Well, homeostasis is an internal balance. And when we talked about homeostasis back in Unit 1, we gave a few examples, one of them being maintaining a constant body temperature of 98.6 degrees Fahrenheit. If our body temperature gets too high, so it goes above 98.6, we obviously don't feel good. If it gets really high, it can be severely detrimental to our health. And same thing if it gets too low. Hyperthermia would be, getting a fever. Hypothermia, like if you fall in a lake, means that your body temperature drops below 98.6 degrees Fahrenheit. Okay, moving on. The word selective permeability is important as we move through the cell membrane here. And the reason it is because selective permeability means that some materials can pass through while other materials cannot. Okay, so let's try and think of a few examples of things that are selectively permeable. I'm just going to give you a few minutes here just to kind of think to yourself, so you can pause the video if you want, and then start playing it after you've thought about it a little bit. So pause the video. Okay, so some of the things, and I'll just list a few just in case you didn't come up with these, but one thing that can be selectively permeable around your house would be something like a screen door. Screen door obviously lets air in, and but keeps bugs out. So that would be something that's selectively permeable. If you make macaroni and cheese on a regular basis, not the microwavable kind, but the actual old-fashioned in a box kind, you know that you have to boil water and put noodles in, and when they get soft, you have to separate the noodles from the water. And most people use a strainer to do that. So that would be another example of something that's selectively permeable. So why would we need to know that term? Well, the cell membrane is said to be selectively permeable because some things can pass through the cell membrane while other things cannot. And what we're going to be talking about over the next few days is exactly how it selects what moves in and out. But again, before we can do that, we really have to understand the structure of the cell membrane, because the structure of the cell membrane really determines what can move in and out. Okay, so let's get to the structure. So the first thing we're going to talk about is the structure of the cell membrane is known as a phospholipid bilayer. Okay, so let's break that word down. The word lipid should be familiar to you because we just talked about a lipid last week. And a lipid is, is a macromolecule made up of 4 different monomers. The monomers are glycerol in the shape, and it's all in the shape of an E, and the glycerol is the up and down part, and 3 fatty acids. And the 3 fatty acids are the horizontal parts of the E. Okay, so that's a lipid. A phospholipid must be a little bit different, so we're going to talk about that here in the next slide. Bilayer, the word bi meaning 2 layers of phospholipid. So there are 2 layers of phospholipids. Okay, and as we move on here, we're going to start talking about what an actual phospholipid is. Okay, that brings us to slide number 2. Okay, so the first thing we're going to talk about is what is a phospholipid. A phospholipid is this molecule here on the left-hand side. And if you take a look at it, you can see some familiar parts of the standard lipid molecule. If you look right here, for example, you've got the glycerol backbone. Let's move this back just a little bit. Oops, I just wrote on it. There we go. Here we go. So if you look right here, it says glycerol backbone. Okay, so there's the glycerol molecule, but it's kind of right here in the middle, which is kind of strange. So it's right about there. That's the glycerol part. So if you want to, you might want to just color code it because it's a little bit different. So you might just want to say, there's the glycerol backbone. Then at the top here, you've got what's called the phosphate group. I like to think of the phosphate group as just a circle. Okay, so I'm just going to draw a circle here. So that's the phosphate group. And then down here, you've got the fatty acids. So it's kind of like, if you look at it, it kind of looks like a stick person without arms. And that's, if I ask you to draw a phospholipid, that's what I'm expecting. Okay, so below it here, I'm just going to draw a phospholipid molecule. You should do the same. So another phospholipid molecule here, because we're going to be labeling this. Okay, so at the top of the phospholipid molecule, remember, you've got the phosphate group, and at the bottom, you've got the fatty acids. Okay, so the top of the phospholipid is said to be hydrophilic. Okay, and the bottom of the phospholipid molecule is said to be hydrophobic. So what is hydrophilic and hydrophobic? Well, hopefully the first part of the word there, hydro, will remind you of water. Okay, and the second part of the word, let's focus on hydrophobic because I think it's a little more familiar to you. Phobic usually means afraid of. Well, the cell membrane or the phospholipid itself isn't going to be afraid of anything, but what it means is that it doesn't mix with water. And of course, we talked about the properties of water. So the hydrophobic end is said to be nonpolar. Okay, and nonpolar things don't mix with water because water is polar. So that's exactly why it's hydrophobic, because it doesn't like water, doesn't mix with water. The top, hydrophilic, is actually polar. And that means that, of course, that mixes with water. So what's weird about the plasma membrane is that it has these kind of two parts to it, where part of it is mixing with water, where the other part isn't. Now, it's kind of hard to see in the picture here, off to the top right, but you have several different parts of the cell membrane, but what I like to do is actually draw it, because I think we can draw it a lot better than what we see here. But just taking a look at some of the parts of the cell membrane in this picture, you can see the phospholipids, and you can see they're kind of hard to see, but the circles are the heads, okay? So the circles up here are the heads. All right, notice there are two layers of those phosphates. Here's some more down here, okay? The legs, which are kind of really hard to see, the glycerol is really hard to see because it's just the way the picture is. But you can see the legs kind of shooting up here, like that, and then these legs down here, like that. And that's why, of course, it's called a phospholipid body, because there are two layers of phospholipids. Okay. So there's that. All right, we've got this over here. You've also got several different proteins around. You can see protein channel. We're gonna just define these and draw these on the next page here. But I at least just want to kind of point them out. You've also got, another protein over here. You've got several proteins here and here. And these are all different types of proteins and have different jobs, and we're going to talk about those. Also, and this is really hard to see, at the top of each protein, so here's another protein right here, there's a little carbohydrate chain that kind of sticks up in the air, and those act kind of like little feelers. When we get to the different types of the different proteins, I'll talk about that carbohydrate chain a little bit and talk about what it's about. The last thing that you see inside the cell membrane is the cholesterol. Okay, the cholesterol, I'll circle them here, are these guys, these little things stuck all over the place, these little chains of little white circles. And again, we're going to go through the functions of all these here on the next slide. So. All right, the last thing we're going to do here is we're just going to draw the cell membrane. We're going to do so here at the top. And we're going to start off with just a bunch of phospholipids. And I find that it's actually sometimes easier just to draw the circles, the phosphate groups, line them up first, and then give the backbone and the legs. And they can kind of crisscross. You don't have to be, an artiste to get this done. It doesn't have to be great. Just a nice visual. Okay, and there's the phospholipids. Notice we have two layers of phospholipids because it's a phospholipid bilayer. The other parts of the cell membrane that we're going to talk about from the previous page, the first one is a channel protein. Go ahead and draw that in here. And I kind of draw it, think of it like a hot dog bun. So, this little middle part's kind of an open space, okay? And things can kind of move in and out through the channel protein. All right, and we're going to go through the specific properties of these here, and we'll get to it in a minute here, but let's finish our drawing. Okay, next up, let's draw what's called a carrier protein. A carrier protein, it looks like a V, I guess. Drawing a phospholipid there to fill that in. And how the carrier protein works is, like, let's say here's a little particle of food that wants to move into the cell. Okay, so let's think of this. The top here is going to be the outside of the cell. The bottom is going to be the inside of the cell. Okay, so we're looking at like a cross section here. And the particle of food can move in, and then what can happen is the whole carrier protein rotates, just like that. And now, just to draw below, you don't have to draw this, but now the V would be inverted, right? It would look like this. Okay, it would be up there, of course, but the little particle of food could then move out. All right. So I'm just going to erase all that. Okay, so that's how a carrier protein works. Okay, let's draw a few more phospholipids here. The last type of protein you'll find is what's called a marker protein. Marker proteins can be up top, like so. They're kind of like, think of them like raisins spread out throughout your cell membrane. And they could be on the bottom. Just drawing a few more phospholipids here. Okay, and here let's draw another marker protein down here. Okay, so those are marker proteins. All right, so let's go ahead and label some of these here. So this is the channel protein, sometimes called a protein channel. Don't let that confuse you. All right, this is called a carrier protein. And these are called marker proteins. Marker proteins also have this little carbohydrate chain, kind of this little thing that's attached to it. So let's label that carbohydrate chain. Okay, think of the carbohydrate chain as kind of like a little antenna, like an insect's antenna. It kind of reaches out and feels for things, and that's what it kind of does. And it can use these are used for cell communication. Remember, cells don't really have mouths. So they need to have other means to communicate with other cells. You can kind of think of these marker proteins as like little messages, like little notes that they can pass back and forth between cells. The cells have to touch, and then when they do that, they can transmit the message. Okay. The last thing that we have inside the cell membrane are cholesterol. Is cholesterol. I'm just going to draw those. I changed my color here. You might want to do the same. Okay, just they're little circles with little tails would be a good way to describe them. Okay, some pictures have them represented differently. Like the picture on the front has just like little white circle chains. And they're throughout the middle of the cell membrane. Okay, they're all over the place in the middle. And what they do, so let's label these as cholesterol. I'll leave it that same color so we can easily pick it out. What cholesterol does is it provides rigidity for the cell membrane. All right, so I'll kind of go through them though. So here we go. Down below, let's write proteins. There are 3 types. There's a marker. Well, we'll go in order. Start with the channel protein. Think of the channel protein as a tunnel where things can just move back and forth. Okay, maybe just draw another example of it right next to it. See, like this little hot dog bun, and it just kind of goes right through there. See, you can just go right through there. All right, 2, that's the carrier protein. I kind of already described how this works. You can kind of think of it like a security door. You know that. So you step into the security door, and then it opens or turns. And then the thing is, whatever wants to move in is inside or outside. It works both ways. So think of it like a door that swings open. Okay? All right. The third type of protein is the marker protein. The marker protein is for communication. You can hear the bell there. So marker protein is for communication. The last thing we have to talk about, again, is the cholesterol. The cholesterol, its job, it's not a protein, so it's different. Cholesterol is to provide structure and support for the cell membrane. The more cholesterol you have, the more rigid the cell membrane will be. Okay. Now, of course, we've heard cholesterol before, and we know that cholesterol is kind of bad for you. Well, if your cholesterol gets too high, it's bad for you because, again, these are rigid molecules, and if they start building up in your bloodstream, they're going to create a blockage because they're hard. And of course, a blockage in your bloodstream leads to heart attacks or stroke and other things. So, all right. The last thing I want to talk about, I'll switch to red, is talk about that hydrophobic and hydrophilic again. If you look at how the cell membrane is drawn, you have kind of 2 areas. You have the inside. If you look up at the picture, I'm just kind of coloring it in. You don't have to do this, but the inside would be the hydrophobic area, right? Because we talked about the legs of the stick figure being hydrophobic. So I'm just going to abbreviate that HP, hydrophobic. You might want to write out the whole word. I'm just running out of room here. The outside, maybe actually, now that I think about it, we're going to have to do more than HP because hydrophilic would be HP too. Maybe just put phobic. Okay, that's hydrophobic. All right, that looks great. That looks all sloppy, but you do better. Okay, so that area is the hydrophobic area. The top, so here is going to be hydrophilic. But notice the other side is hydrophilic too. Okay, so that area is going to be hydrophilic, so is this area. So we got to kind of draw another arrow here. This is also hydrophilic. So the areas, so inside the cell membrane is hydrophobic. Outside of the cell membrane, it's hydrophilic. Okay, so that's kind of talks a little bit about how that works. That's why we can see it. It almost looks like a layer of something that's not dissolving in water. The tops and bottoms can dissolve in water. Okay, and that's the structure of the cell membrane.